So hello to everyone and welcome to the first Pharmacelera scientific seminar on drug discovery on new antibiotic in the AMR era. Let's say traditional antibiotic drug discovery pipelines mostly arise from the screening of libraries of small synthetic molecules or natural products, or from scouting traditional medicine, traditional anti-infective medicines. Both have produced the drugs that we know, and that led to in the past to the known mechanism of action and to a myriad of second and third and fourth generations of improved analogs and families. This massive screening has limited drug discovery potential related with the available synthetic and natural chemical space. That's the limit there. More recently, we have focused on finding small molecules that inhibit a certain essential protein. This focus on the lethal protein inhibition has also limited drug discovery potential based on the availability of such essential proteins to be targeted and of known recipes to transform this protein inhibitor into a bug killer and further to an antibiotic. Okay, it's only recently that disruptive new technologies to find new antibiotics, like for instance bacteriophages, microbiome, pathogen selective antibiotics, gene editing CAR-T, and so on, have opened new avenues that will show soon their potential to generate new drugs, but they are still on the incubation. We have today a great panel of scientists that have been scouting for new antibiotics beyond the limits of currently available space on chemicals, targets, and available technologies. And the intention of this webinar is to discuss and hear their opinion and learn from their experience to what extent the current approaches are exhausted and the only option is to jump to new disruptive approaches, if available, or if there is still room to find new antibiotics by tweaking and updating the traditional approaches. I am Albert Palomer, and I shall be your moderator of today's webinar. I am pleased to introduce the expert panelists, and I encourage you to read about their accomplishments in the fields. We have Dr. Eric Braquet, previously at the Sanofi site in Toulouse, running several antibiotic discovery programs, and now at Evotec, probably the largest pharma company effectively involved in antibiotic programs. We have also Santanu Datta from Bagworks, a US and India company, very active in the field, that has been recently awarded by Carbex a grant to develop a new broad-spectrum gyrase topoisomerase antibiotic. And finally, Mark Gitzinger from BioVersis, a Swiss company that is promoting an antitubercular candidate to clinical development and has also been recently awarded a Carbex to support the development of new first-in-class antivirulence small molecule designed to disarm bacteria. Before we jump into the conversation today, I want to let our audience know that if at any time during the webinar you would like to ask a question, feel free to use our Q&A tool. We'll make sure to reserve time at the end of the answer to answer the audience questions, or to answer these questions after the webinar if time runs short for us. Okay. Now let's thank Eric, Santanu, and Mark for their time and dedication. Today we shall discuss the limits of AMR drug discovery, and now we'll move to hear from our panelists first their opinion on the boundaries of the current synthetic and natural chemical space, then thinking about the target space and availability of the druggable new targets. What are the limitations, since not many new targets have been drugged during the last decades? And finally, their view on the new technologies arising in the field, and we'll start hearing the opinion from Eric. So please, Eric, we'll give you words and to hear your opinion about these topics.
Thanks, Albert, for the introduction, and good morning to everyone. I'm Eric Braquet from Evotec ID Lyon. So in the next few minutes, I will give you a quite general and educational introduction to the topics and questions raised by the webinar. Next slide, please. Let's start by introducing the two key approaches of finding in antibacterial drug discovery, namely the target and the phenotypic-based approaches. So when I mean phenotypic, I mean here growth inhibition, so looking at the inhibition of the growth of bacteria. Next click. So these two key approaches have largely failed in the recent decades, and they have not delivered on brand new marketed classes. This is for a number of reasons. I will come back to this later, but first I can say that for growth inhibition screening, generally they don't just afford valuable hits, especially active versus gram-negatives. For the target-based approaches, they have not fulfilled the promises of the genomic area. I can remember 20 years ago that just after the first sequencing of the first bacterial genomes, that we had the impression that there was a wealth of new targets awaiting us, and this just didn't happen. So the consequence of this is that we have today few new antibacterial scaffolds to work on, few new chemo-validated targets, and so the consequences is that the search for new antibacterials tend to concentrate on well-validated targets and on non-classical non-classes and scaffolds. Of course, this has resulted in some very significant success recently, but you understand that this is not the best way to overcome all pre-existing resistance with non-classes of antibacterial. Second slide is about the chemical matter. This is an endless discussion between chemists and biologists, and this is really central. What can be concluded from the literature today is that the usual screening collections are not adapted to find novel, valuable antibacterial starting points. And this has been clearly demonstrated by Mosers in 2008. The Fischem space of the antibacterial compound is very different from the usual Lipinski space of pharma collections. And this is no surprise. The pharma collection compounds have not been designed to cross or to penetrate the complex membranes of bacteria. This is even more true for the more polar anti-gram-negative compounds. In parallel, the access or the identification of new natural products have also decreased largely. And so the conclusion is that indeed there is a pressing need to find more adapted chemical matter to find new starting points. Of course, the question is which chemical matter? Probably, in my opinion, natural products are still one of our best chances to find novel antibacterials. But of course, you have to make things differently. Otherwise, you will end up with the same compound that are isolated again and again. So you should make things differently. Probably you need to go to some new exotic producers. You have to try to culture the unculturable producers. You may apply some bioinformatic and genomic approaches. You can try to awake the silent biosynthetic gene clusters. And all this should be also accompanied by optimizing the screening format, the deconvolution process. So a lot of things that make this is a challenge and a lot of investment. Of course, you can try to revisit some old underexploited classes of antibacterials. Now, there are some recent success along these lines, and this is a good way to have a good starting point. You can try to evolve some anti-gram positive only compound into gram negative compounds. Again, there are some examples recently. If you think that diversity is a problem of the chemical matter, you can go to some large libraries made by the DNA encoded or the diversity-oriented synthesis approaches. You can go for antibacterial specific small molecule collection, but I'm not aware there are so many. And of course, you can also try some chemo informatic approaches. And there is a recent paper on the discovery of allicin that make the buzz recently. Next slide is about phenotypic screens. The good point about phenotypic screens that by definition, your starting point, your hits, have whole cell activities. Of course, they have also a unknown mechanism of action. as I told you already, you can expect from these approaches to have maybe some anti-gram-positive hits, but very few valuable anti-gram-negative hits. Another advantage of the phenotypic screening approach is that you may uncover some new targets. This is good, and this has been particularly effective in the field of tuberculosis, for instance. Next one. But here the key question is that are the MIC, the antibacterial activities that you see, are really specific? There are many ways to be toxic to a bacteria, but only a few ones are really selective to bacteria. So the challenge is really to demonstrate that you have specific antibacterial activities, and for this you need to set up a suite of assays that are designed to eliminate the junk hits that are several, numerous. So in general, this can be, of course, cytotoxicity. This is very classical, but you can also have a look on chemical reactivity, mitochondrial toxicity. This is maybe less known. Membrane perturbation, that is really a pain in the neck. Hemolysis as well. This is good to have very early clues on mechanism of action, so you can do so by molecular, macromolecular synthesis, cytological profiling, looking very rapidly at mutant resistance and sequencing. So this really can help you to see that you have a specific mechanism of action. And of course, you should also evaluate very rapidly cross resistance and frequency of resistance, because you don't want to discover late that you have a major problem of resistance. Next. So just a few recommendations. For me, probably there is a need to change the bacteria and or also the way you interrogate bacteria. I think this might be in some instances more fruitful than looking for some different chemical matter. There are many options in this scenario, all coming with challenges, of course, but I think there are maybe possible low-hanging fruit. Of course, rather than wild-type strain, you can use efflux or influx engineered strains. You can go to hypomorph, so you have down express some genes so that the bacteria become very sensitive to inhibitors of the gene products. You can have some biosensor strains that detect some I don't know, for instance, defect in the membrane or in the cell wall. You can go also to some underexploited species, so looking for compounds that are Acinetobacter or Neisseria specific. This has not been done really systematically or just starting. You can go to different lifestyles, and I mean by this, for instance, biofilm, dormant forms that are really also important forms of bacteria. You can try to have different culture conditions, and for instance, you try to screen into a condition that are more physiologically relevant, the in vivo mimetic medium. Of course, you can also imagine to go to whole animal models, for instance, say elegans, the worm. Next one. So last one on the target-based approaches. So I would like to stress that in this approach, there is many risks and traps along the road. First message is that finding and fully validating new targets is very difficult. So this is a question of essentiality. Yes, this is true, but also vulnerability. So do you need to inhibit 50% of the target or 19.9% of the target to have really a clear effect? This is also, of course, a problem of selectivity versus the human orthologs. Drugability, this has been often belittled by biologists. You should also look at the resistance risk, and so if you have a low fitness cross mutation leading to easy emergence of resistance, ubiquity if we are looking for large spectrum. Of course, you may have problems because the social requirements that are needed for target affinity are not those needed to penetrate the bacterial membrane. So this is a special case of undruggability. You may have problem also because this is very recurrent. Pharmacophores or characteristics necessary to penetrate are associated to tox or physical issues. Next one. Just a provocative question. So when you see that there are been so many failures in the past, you can ask you the question that, are there so many new relevant targets around? So this is really the question. If you don't agree with this question, you should answer the next question, which is, why don't we find more of them by phenotypic screening? The typical risk and traps for this approach is that you have potent target inhibition, but you have no antibacterial activity, no MICs. And this is in general due to the problem of penetration, influx, efflux. The other trap is that you are not optimizing your MIC versus the target, but you are just optimizing versus membrane perturbation. So recommendation, next one, click. So you really need to have tools to demonstrate that you are still on target activities. So you may have overexpressors, hypomorph, resistant mutants, macromolecular synthesis, to be sure that what you are optimizing is still on target. Of course, you should also try to understand the reason of your poor antibacterial activity when it is the case. And this is good to use influx and efflux engineer strengths to really decipher the contribution of influx, efflux, target affinity on the poor MICs. Of course, you can go to non-targets, but provided you are targeting the binding site, and we have many recent examples with topoisomerases, ribosome, cell wall, etc. You can go for ultramembrane proteins or periplasmic targets that don't have compounds, inhibitors that don't have to cross too many membranes, but there are not so many. You can apply permeability guidance that have been predicted recently, and finally you can try to adjust the active transporters to improve the influx, and you know that probably the siderophore conjugate has been very popular lately with some key success. So that's it for me. Thanks for your attention.
Okay, thank you. Thanks a lot, Eric, for making this summary of and to center what is the problem really, and some options of solution. That's very challenging and motivating. So now we would love hearing Santano's opinion on these topics, targets topics, essentially. Okay, thank you, Santano.
Hello, hello. This is Shantanu. What I will do is ask the questions which have plagued the antibiotic drug discovery, and try to give you a glimpse of some probable answer. The first slide, the next slide. Can I get the next slide? Yeah. Now, what is wrong with the current, not current, I would say the last 20 years of anti-infective drug screening is, in my view, that the drug discovery moved, unfortunately, from the hands of chemists and biochemists to molecular biologists like me. With the advent of genetic engineering, whole genome screening, we could sequence nearly all the bacteria, pathogenic bacteria, and then identify essential genes. Now, there are, say, for example, if you take an E. coli, there are 4,300 genes, of which 1/10, around 400 essential genes. So, as a target hunter, you could say you have 400 possible targets. You take up an essential gene, set up an enzyme assay, and set up an inhibitor screen. Now, all the big pharmas, including where I was in my previous avatar, we tried this and it was a big failure. Can I go to the next slide? So why there was this failure? The failure was because we assumed the very simple equation which is written in the bottom of the slide. That means a genetic knockdown is equal to a chemical knockdown. We assumed that if a gene can be knocked down, or if it can be reduced in its expression level, the compound inhibition would also do the same. Now, this is a very, very big assumption, and as I will tell you, and I think my previous speaker also alluded to it, that all enzymes are not vulnerable to inhibition. What does it mean? It means that if you knock out the gene, the expression level is 0, but when you do a chemical inhibition, you are not making it 0, you are making it intermediate. And that intermediate level is good enough for the cell to survive. Now, we assumed also that vulnerability of a gene can be estimated by genetic knockdowns. That means if you put the essential enzyme under an inducible promoter, you could measure the vulnerability of the gene. But however, this is not true. If I go to the next slide, it is very clearly seen that there are various kinds of essential genes. Say, for example, you take 3 genes, A, B, and C. The gene A, which is essential, when it is knocked down, that means when it is in a KO status, maybe a 50% expression lowering will bring the growth rate from 100% to 0. The essential enzyme of class B may require a 10% residual activity to bring the growth rate to 0. And if you are targeting the class C, which is, I think, the most of the enzymes are, you may require to inhibit the enzyme nearly 99%. That means you have to knock down the essential enzyme 99% to bring down the growth rate to 0. So you see in this case that essentiality is just a starting point. It is not the absolute requirement for a target. Now, if I go to the next slide, I will see that there is a general understanding that drugs or inhibitors mainly target the key enzyme and reduce its activity. Now, inhibition in classical, graduate textbooks of biochemistry will tell you that there are 3 different types: competitive, uncompetitive, and non-competitive. And in this table where I have put the 3 types of inhibition, in addition to that, I have also used the knockdown, where the enzyme levels are put down by inducible promoter. You can see that all the different types of inhibition have different effect on the kinetic parameters. So the enzymes generally have 2 primary kinetic parameters, the Km and the Vmax. So the knockdown basically does not have any effect on the Km, but the Vmax, which is the concentration of the enzyme into kcat, the kcat remaining unchanged, but the expression level is going down, so only the Vmax goes down. In the competitive inhibitor, on the other hand, because the inhibitor is competing with the substrate, the Km is increased, the Vmax is unchanged. In uncompetitive, similarly, both Vmax and Km are decreased, and in non-competitive, it is the Vmax which is decreased and the Km is unchanged. So which means that chemical inhibition is not equal to genetic inhibition. And finally, because we are looking at drugs which will be chemical inhibitor. Now, if I go to the next slide, I will see, I have mentioned earlier that there are only 400 essential genes which we thought were possible antibiotic targets, but only less than 10 are clinically validated. The question is why? If you look at the validated targets, that means the inhibition of protein synthesis, the inhibition of nucleic acid synthesis, the inhibition of cell wall, there are membrane disruption agents like daptomycin and colistin, or there are only very few, which blocks metabolism, pathways like THFR inhibition by trimethoprim and sulfamethoxazole. So most of these targets are basically which synthesizes the macromolecules. There are hardly any, except the DHFR inhibition by dimethyltryptamine. There are hardly any enzymes. There are many essential enzymes, but there are hardly any small molecules which act as an antibiotic against the general metabolic pathway. And the reason is simple. Metabolic pathways are substrates which are converted to enzyme, and most of the inhibitors are generally competitive inhibitors. And when you have a competitive inhibitors, especially in a linear pathway, the moment you inhibit the enzyme, the substrate goes up. And so, as I said, if the substrate goes up, the inhibition goes down. So not only is the vulnerability an issue, the type of inhibition that is used is also an issue. And that is the reason why many screenings yielded fantastic nanomolar inhibitor, but it did not do anything to the MIC. That means it did not kill the organism. If you look at the next slide, you will see that one could argue that the compounds did not also inhibit because probably they were not entering. That is the impermeability. And that is the case with many compounds because of the chemical space that we're in. And currently we have some understanding how to make molecules to enter the drug. And there are other ways like pumping out the efflux. There is most of the gram-negative bacteria have very high efflux pumps, very high efficiency efflux pumps, which throw out the enzymes. And there are inactivation processes which inactivates the enzyme, inactivates the drugs, which we all know, like beta-lactamases and so on. So if I go to my next slide, which is the last slide, I will explain what we have done is that we have understood that the single target is not a good idea because it will have resistance phenomena. So we have targeted two essential genes in the validated target, that is the replication, that is gyrase and topoisomerase. We have these molecules which are called gyrox molecules. And one of our lead molecule 977, here in the pictorial, we show that it works against multi-drug resistance bacteria. And the reason why this is so far, it has been successful in test tube, in animal, and also it has gone through the GLP tox package, and it's waiting for the phase 1 clinical trial in the next few months, is that it is hitting two targets with equal potency. When you hit two targets with equal potency, that is gyrase and topoisomerase, then what happens that a single mutation cannot change the MIC. You need two independent mutations. And since you need two independent mutations, the probability of two mutations happening simultaneously is astronomically low. We cannot, in the test tube, in the lab, if we play 10x10 bacteria, we don't get any resistance. So far, we have not been able to get resistance against this dual target, which I think is a way to go for the next generation, where if we can fit in two targets which are already validated and make inhibitors against a common inhibitor against the two targets with isopotency, I think this will be I won't say straightforward, a more validated way to move forward. Thank you.
Okay, thanks a lot, Shantanu, for putting in order the business of the targets, the the vulnerability and the mechanism of actions and what is worth and what is not worth there. So thanks a lot. And finally, now it is time for Mark to share his view and experience on these topics. Please, Mark, go ahead.
Thank you very much, Oscar. So hello, everyone. I will actually now give, after my previous two speakers, very practical example of a project that really benefited not from phenotypic versus target-based screening, but actually from the combination of both of them. This is an example of tuberculosis. And next slide, please. You know, one of the technology platforms that we have in Bioversus in our company is actually targeting transcription regulatory or transcription regulators with small chemicals. We call this trick therapeutics. And the basic or the rationale is described here. This is obviously a new target series in bacteria, has been tried a couple of times. And it is obviously key to identify the essential pathways that actually really make a difference then in a therapeutic setting. We have on the left-hand side the tuberculosis example, which I'm going to present to you, where you see that we use the target of transcriptional regulators to overcome resistance against existing antibiotics, while in the other programs that we follow on this technology platform, we're actually targeting virulence and toxin production, for instance. Next slide, please. So tuberculosis, just as a small background, we talk a lot about AMR, antimicrobial resistance, and focus often, of course, on the gram-negatives. Not to be forgotten is that TB is still the largest actually bacterial infection killer today, has a lot of resistance, and overall a quite unsatisfying regimen because it's very long for even drug susceptible TB bacteria. It's a global problem, of course, more focused in the developing world and in the BRIC states. Please, next slide. One of the particularities in TB is, and I think this was never actually intended by anyone screening for new drugs, but you find that from the old drugs up to the very new ones, which is Delamanid from Otsuka, one of the two newest approved anti-tuberculosis drugs, a lot of the tuberculosis drugs are actually prodrugs, and not in the sense of being a prodrug for better bioavailability or something in the human body, but actually they enter as a chemical compound, the tuberculosis bacteria, and then modified in kind of a suicidal manner inside tuberculosis to a final product that is then killing the bacteria with different targets, often in the cell wall synthesis. This concept is interesting because it also makes a complete new change or difference in how TB develops resistance to such drugs. And what you see in the field is that the resistance against most of the prodrugs is, of course, not efflux, because once the drug enters the bacteria, it's directly modified into a much larger molecule. But it is actually in the enzymes to bioactivate the prodrug inside TB. So that's where you face resistance against isoniazid, ethionamide, even delamanid already, pyrazinamide, etc. And that's where the clinical relevant resistance comes from. So we took this approach looking at ethionamide, a very old drug. In the most of the Western world, ethionamide is used as second line still today in resistant TB patients. And then towards Asia, it's a molecule called protonamide, but basically they work exactly the same and they're the same kind of chemical class. Now, we looked at the bioactivation pathway of ethionamide and identified one of the transcription regulators that down regulates the enzyme that is responsible for the bioactivation. And this was our hypothesis and working hypothesis for the target-based screening. And while I fully agree with Santanu that a knockdown of a gene is never directly equal to a chemical inhibition. In this very instance here, it actually worked very well that when you knock down the transcription regulator ETHR, you have actually a full activation of ethanolamide and a much more effective drug than if this repressor is there. So our hypothesis was to prevent this transcription regulator from inhibiting the expression of the enzyme. That was a target-based approach. We used that for this, a very simple thermal shift assay where you measure the temperature difference if a compound binds to the target or not. We also gave into this equation the DNA, the known binding site for the transcription regulator. But in parallel, we did a second, much more sophisticated screen in a high throughput manner, where we actually infected macrophages with fluorescent tuberculosis bacteria and used sub-inhibitory concentrations of ethanolamide, the drug that we wanted to kind of recover or make more efficient, and screened our chemical library also against this screen in a high throughput manner with the OPERA system. And then next slide, please. What came out of it when you plotted the two results of the screen actually against each other was this very interesting picture. So on the very right-hand side, the oxadiazole series that we identified played exactly along the hypothesis. We have a good target-based inhibition, which you see on the x-axis, and we also have what we call an EC50, an effective concentration on the infected macrophages, without, by the way, killing the macrophages. So this was another aspect of the screen that was very helpful, having directly a kind of tox marker. So this was perfectly fitting the hypothesis, and everything was great. We were very happy. But of course, we had one downside. There's sometimes also in the field, this was only actually learned after we started this project, that you sometimes have patients that develop, or TB bacteria that actually develop stop mutation in the enzyme. Of course, then it doesn't help if you have a broken enzyme to use the or to block the transcription regulator. So instead of being the end of the project at that time, because we couldn't actually come to an MIC 99 with our approach, we went back to this little plot here, and we started to identify, obviously, that we had compounds, the Spiro series, for instance, to the very left-hand side, which were quite extreme, if you want to, because we had absolutely no target inhibition or interaction. But at the same time, we had a very strong effective concentration on the macrophages. And if you then go, or on the infected macrophages, if you don't go to the next slide, please. What you see here is a schematic. Some of this data is already published. So the ETHR2 pathway is published in Science, and then we have published the original pathway and hypothesis with ETHR. The blue one in Nature Medicine, and the first development of these compounds. What is currently under or submitted is actually the newest pathway, the MIM-A-VRS pathway, which is, as you see, by the different classes, which one is hitting these pathways. So the Spiro series is on the ETHR2 and VRS MIM-A pathway, piperidine series as well, while the original series, the oxadiazoles, are only on the ETHR. And the beauty of this is now that we have a program which resulted in very advanced lead compounds. Actually, one we expect actually to start to dose first subjects in October this year in the first clinical trial, where we have now compounds that are super effective in making ethanolamide, an old drug, much more effective than it ever was, even in wild-type strains, and at the same time overcome all the existing clinical resistance that exists, which lies in that blue pathway, ETHR, and then the enzyme ETHA. And this gives us back the full potential for the first time of ethanolamide, a very effective drug with a proven pathway, and makes this drug really available again. It's a very unique pathway in TB. which allows for very fast killing and bactericidal activity, especially with this high effective concentration that we now have of thionamide. Next slide, please. So I think a couple of things to sum up this presentation is actually, we heard from Eric in the beginning, when you do different kind of screens and media and so forth, you potentially look into awakening silent pathways. I can tell you that the ETHR2 pathway, we still haven't found a stage of the natural TB bacteria where this pathway is active at all. It's only our molecule that activates it. For the MIM-A, it's also a fairly silent pathway in most of the TB status of its life cycle. So we really do awake here silent pathways. We have the benefit of making of an old prodrug a significantly more effective product that even is safer now at the lower doses we can deploy, and again, more effective in overcoming the resistance. So this is really a cool combination that we were able to identify here, with combining target-based screening from a hypothesis, but at the same time being very open eyes in phenotypic screening. And I shall also say that what you see on this slide here, all of this effort, especially in TB, wouldn't have been possible with a very, very well functioning public-private partnership. And I think this is anyway, the new reality in antibiotic resistance development or drug development, that we have to partner. It's complex pathways. Money is not hugely present in this field. So having partnerships is very beneficial, and led us here to, in a long process, to really a completely new hypothesis, drug target, and possibility to have a new combination now being assessed in clinical trials. And with this, just out of courtesy, of course, mentioning the different people or the main people that were involved in this program, you see that the partnership is obviously with the public side, the centers in Lille, in northern France, but also with our pharma partner GlaxoSmithKline. So thank you very much.
So thanks a lot, Mark, for your very clear presentation on your approach to the TB, and it's very interesting public PPP structure that is helping you move these drugs forward. That's a very interesting organization, really. So now we are on time for questions from our audience, and I will try to summarize some of the questions that came out quite often. One of the questions relates to the knowledge and experience of people, and that relates to the two different areas, natural products and development. So, for instance, natural products have been the most efficient sort of new chemistry in the field, but now most companies and the knowledge and expertise in the hands of their scientists have disappeared. And that's something that appears quite often in all the surveys by AMR Initiative or GARP, that the knowledge and experience has disappeared. So few natural product companies are, and their experts remained alive. And moreover, they usually have limited muscle and prefer to dedicate to oncology rather than to AMRs, despite there is this high unmet medical need. So in your opinion, and that's a question I understand for the three of you, is a good strategy, is a worth strategy to recover the knowledge and experience, that means the people, to natural products chemistry? Who wants to start?
Maybe I can start. Please go ahead.
Yeah, go ahead, Eric.
Okay. Yes. So for sure, the dissipation or the disappearance of expertise is a critical point. This is probably true, but up to a point, because a number of small biotechs have integrated the experts from big pharma when they were laid down. So I think this is partially true, but this is something that is critical. For instance, I think that we should really foster all opportunities to gather young researchers with the more expert ones. So this webinar is a good illustration. So this is a way to transfer our knowledge, of course. What is also, in my experience, working quite well is also the consortium. So I happen to be the FPR co-coordinator for the Enable IMI New Drug for Bad Bug consortiums. And one of the key objectives of this consortium was at the beginning to really, if I quote correctly, educate the next generation of antibacterial drug discoverers. So this is really something that is important to keep in mind. We have to educate the young generation and to keep the knowledge alive for the next generation.
Hello, this is Shantanu. I will try to give you a personal experience. For 20 years, I worked in AstraZeneca in Bangalore, and we had a great anti-tuberculosis program. And in 2014, the organization was shut down, and Bugworks was formed. Though I had left early, Bugworks was from the best of the talents that were available in. So the core talent in Bugworks are from industry, our chemists, our biologists, our microbiologists, our animal experts, they are all from AstraZeneca. So I don't think it gets lost. Also, what happened, many of the top experts, they start their own companies, and that is what has happened with many big companies when they close down their anti-infective program. And sometimes small is beautiful, and small companies can very rapidly pivot. One of the guiding lines is that fail fast, fail cheap. In a big company, there are multiple layers of administration, which in small companies are not. So they become more efficient. So I am, from my personal experience, I think there are pros and cons of this. This is not a one-way negative.
Yeah, I mean, if I may add, I quickly take off my head as Bioversus CEO and put on the head as representing Beam Alliance, which represents quite a lot of SMEs in Europe. And I have to admit, I am concerned with the loss of talent and knowledge in the field due to the very challenging funding situation in antibiotic drug discovery and development. We cannot deny that it has become nearly a task impossible to convince venture capital to come into the space at these days, which has all been triggered by some of the bad news from US companies that came onto the market. I think it's a very complex topic, but bottom line is we have lost some talent. Luckily, just as Shantanu says, and also Eric, there is initiatives from the public funding side to help and to continue to educate people. And luckily, we have some of these experts that truly believe in our mission and what we do, and actually don't give up and form these small companies. So that's a hope, and it's great. The fear I still have is that if we don't have significant change coming up in the not too distant future, we will actually lose even some of the small and beautiful setups, and that will be very sad. So in this regard, maybe some advertisement for this afternoon, the announcement from an AMR initiative. I think at 4 or 5 or something, the press conference is, that might be helping. And then Oscar, on your question for the natural products, I think every approach is currently worse to give it a shot. Natural products have proven to be very successful in the past. At Bioversus, we have more focused on small molecules and synthetic chemistry rather than natural products in the past. But in the meantime, we actually also have natural products in the pipeline. And I think it is just worth give it a shot. The beauty of antimicrobial drug discovery is that in the preclinical phase, which is arguably the cheaper phase, we can actually do so many good testing and validations of our models, etc., to understand if the approach is moving forward, that we can de-risk a lot in the earlier stages. Sometimes we're asked why it takes so long, but we can do a lot in the earlier stages before we go then into the clinical, more expensive development. So I'm certainly pro giving it a try, and database then looking at what works.
Albert, maybe I can also comment on natural products. So I guess, so we have, when we were Sanofi and now still Evotec, we have strongly invested in natural products because we were believers that really this was the best chance to find new scaffolds. So we had a number of, we had a strong collaboration with Fraunhofer Institute in Germany, with also the Elmos Center Institute also in Germany. So we have invested a lot. So we had some lessons learned and some that were not nice. But probably what is really stemming from this effort is really the need for new producers. And new producers mean new chemistry. And so some are already visible in the literature, but if you look at some exotic producer like Myxobacteria, Xenorhabdus, you can find new antibacterial that are not like at all on what is existing. So I think that really this is a really worthwhile investment. But of course, as I told you in my presentation, this is a quite expensive one because you need to have the expertise, yes, but you have to have all the facilities. You have, for instance, also to have a very strong direct application database. So meaning thousands of compounds that have mass spectroscopy data. So this is really a huge investment, but if you are able to do it, I think this is a sensitive or smart investment, yes.
Great, thank you then. I see from you three words that although this is quite difficult and expensive and some expertise has been lost, but if a young scientist approached to you, you would suggest him or her to join the antimicrobial research. It's still worth, and there is since there is the unmet medical need. That's clear. Another question relates more to the use of computational tools. Let's say traditional computational tools, in fact, have had little impact in antibiotic discovery. But recent works by Richter, by O'Shea, and so on, have opened doors to better understand some bacterial processes and use them for computational prediction. How do you see the use of such tools to design better and more efficient chemical libraries, or to modify and make sub-libraries of what we have now so that they are tailored for antibiotic discovery? Whether these libraries are small molecule chemistry or natural products, but that they are tailored for antibiotic discovery. What do you think of that?
Yes, so this is again an important question. So I think that you mentioned libraries, but also I want to mention just optimization. So you have to guide your optimization. In general, you have always problem of penetration in flux efflux. So you need to have some guidance, some rules to help you optimize your antibacterial activities. And you are true that since maybe 10 years or a little bit more, that we have more food for thoughts. So we have some general guidelines. So you mentioned O'Shea and Moser work in 2008 that really set the scene to demonstrate that antibacterials were really different and giving some clues. I can also name the work from AstraZeneca, Brown, that to make a comparison between the historical antibacterial hits and the rest of their collection. You have also Manchester that published some guidelines about efflux. So all in all, and of course, much recently, the guideline from Ergen-Rover that makes stress the importance of globularity, the presence of an amine, primary amine, and capacity to address gram-negative bacteria. So all in all, this is moving. We have also some more also coming on the modeling of the proteins that are central in the penetration of gram-negative compounds. So this is coming, and not only for libraries, but also just to guide your optimizations. This is really key, and I'm sure this will develop because we are really needing this kind of good, guidelines because we have always the same kind of problems.
Medicinal chemistry is central to drug discovery, and in our quest for a good antibiotic, we continuously use computational tools like in our Gyrox and other programs. We have used computational tools to make compounds which are defiant of efflux. We know the structure of the efflux pumps, and we try to make molecules that don't bind to the efflux pumps, and that requires a lot of computational things. So there are multiple computational tools, and not only 1, 2, 3, there are so many computational tools. So the medicinal chemistry is innate, intuitive approach plus the computational tools is absolutely central to modern drug discovery, especially in antibiotics.
I mean, there's not much to add other than to say, sorry to be very blunt, but we all know the saying "shit in, shit out," and this is really true in the models, I guess. The point is they are very important to be consolidated and used during a proper drug optimization and potentially even discovery program, but make sure that you ask the right questions to the model and that you try as much as possible to learn from the available knowledge that is already published, and also from your own experience how you can actually continue to inform the model to really help you. And then I think it's a matter of using the information from the model, and I guess what Santanu also alluded to, then biochemistry and experimentation, see if you're on the right track and if it can be validated.
And probably artificial intelligence is going to help, I guess. We have some internal experience with deep learning models to predict MICs, and so I think this is going to develop.
Mm-hmm.
I think we got 3 panelists that they all 3 are in favor of using properly these tools. So
yes, properly. Yes,
it's clear. Properly. That's in capital letters, let's say.
Yeah.
Okay, that's great. So we are shortly approaching the end, but I would really like to make you a general question for the 3, but very shortly. We have seen that there is a lot of energy and movements to promote filling the pipeline. We see this afternoon, this AMR initiative and so on. But at the end, the main question is this broken market. Do you see that finally there will be some solution to the final market, which is at the end the final definition for the investors, for the big pockets, for the deep pockets to invest in antibiotic research? Do you think that there is, in short or medium term, a solution to the market perspective?
Yeah, if you want to, I can go first in this one. So I'm generally an optimist, and I think really that we have movement. The problem is that we need novel political guidelines or guardrails to establish a fixing of the reimbursement situation. And once it goes into the political sector, timelines are extremely difficult to predict. So this is my biggest worry. I'm actually convinced that we will have change over time. I also think that while I really don't want to make any use of the suffering that we have worldwide due to the COVID-19 pandemic, but I think what it did show is that infectious disease do matter, and how much infectious disease have actually an impact, not only on the health of a few people that is, potentially ignored by some other decision makers, but actually an impact on our entire lifestyle, way of life, and economy, everything that is around us. And I think this potentially actually has helped to open the eyes to see that preparedness is significantly cheaper than repairing the situation right now for all the governments globally. And this is my hope, that we can now convince governments and decision makers of the G20 that preparing for the AMR pandemic by actually investing into a healthy and not exaggerated, but healthy market environment would make sense. So I am optimistic. The initiative of today will be an intermediate relief as far as we know, but it will certainly not replace a working market environment. So I think nothing can actually replace this, and we need to, all of us that are in the field, constantly advocate for this with every politician that we know.
Thanks, Mark.
I totally agree with this. In addition, I think we need a very rapid diagnostic, which is still not there. Maybe a 15 minutes, 30 minutes diagnostic, which will identify the bug and the AMR pattern. If that is there, I think in addition to the government, the thing will move forward. I think technical people are still, that diagnostic is still a big hole. So especially in countries like India, where broad spectrum antibiotic are given straight away in ICU, where the doctors don't have time to look at the AMR possibility.
Yeah, so I agree with the previous speakers. So I think that something has to change for sure, because the business model is just broken. This is the expression that is used. And Mark alluded to with this recent example of US companies managing or being successful in registering new compound and just going into bankruptcy in the next months because they were unable to have the sufficient money to sustain the post-approval work. So this is just crazy. So there is a big medical needs, but there is no market today. So something has to change. So, and for me, so this, we have been saying that for years. Things are moving, yes, but still slowly. I think that probably the one part of the problem is seems to be well handled. This is the part of financing the research, so the so-called push incentives. So there are a lot now of funders like Carbex, Novo Repair, etc., that can really help the early stages. But for the end, we still need what is called the pull incentives. And there, this is really in the hand of the politicians, so it's more complicated. But we think we are seeing things moving. There are some recent pilot studies, I would say, from UK and Sweden that are really going into the right direction, trying to support right from the start the company coming with an innovative antibacterials to give some over 5-10 years to give some regular revenues and to delink the volume to the reward to the innovator. So this is really the key points. And John Wex, for instance, has been the key advocate for this kind of strategy. So this is the kind of things that we have to make differently to have a chance to work, because today this is not sustainable, and that's it. And we cannot manage this as potential ill people.
Okay, great. Thanks. We know the challenge is there, so let's keep optimistic, and we also know things are moving forward. That's clear. It's an interesting and unmet medical need. This is the case. So we are slowly reaching the end of the webinar. Unfortunately, some questions could not be answered. We'll try to answer afterwards by email. I'll take this final occasion, to thank Eric, Shantanu, and Mark for sharing their experience, and to all the audience for attending the webinar. Just finally and briefly, we would like to remind you that the next Pharmacelera webinar on practical cases will be on how to increase the hit diversity, how to get more and more diverse hits if possible, using PharmaScreen. We hope it will also be of your interest to the audience. Okay, thanks again, and look forward talking soon. We all hope that this seminar were of interest and help all of you. Thanks a lot.
Thank you.
Thanks a lot.
Bye.
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 Bye-bye.